video. In this video, we are going to see about HeLa cells. HeLa cells were the first human cells to be kept in culture for long periods of time and are still in use today. Unlike normal cells, which have a finite lifetime in culture, these cancerous HeLa cells can be cultured indefinitely as long as conditions are favorable to support cell growth and division. HeLa is an immortal cell line used in scientific research. Before seeing what is immortal cell line, we'll see what is mutation, cell division, and cellular senescence. Mutation. Mutation is an alternation in the nucleotide sequence of the genome of an organism. Cell division. Cell division is the process by which a parent cell divides into two or more daughter cells. Cellular senescence. Cellular senescence is a phenomenon characterized by the cessation of cell division. Now let's see what is immortal cell line. An immortalized cell line is a population of cells from a multicellular organism which proliferate indefinitely due to mutation and have evaded normal cellular senescence and can keep undergoing division. The cells can therefore be grown for prolonged periods in vitro. The mutations required for immortality can occur naturally or be intentionally induced for experimental purposes. HeLa cells are termed immortal in that they can divide an unlimited number of times in a laboratory cell culture plate as long as fundamental cell survival conditions are met, that is, being maintained and sustained in a suitable environment. There are many strains of HeLa cells as they continue to mutate in cell cultures, but all HeLa cells are descended from the same tumor cells. The HeLa cell line was developed in 1951 from a particularly aggressive strain of cervical cancer cells taken during a routine biopsy from a 31-year-old mother of 5, Henrietta Lacks. She was treated for the disease by Dr. George Gay. As the head of tissue research, Dr. Gay was at that time attempting to establish immortal cell lines that could be used in medical research. The cell line was named HeLa after the first two letters of Henrietta Lacks. Dr. Gay's lab assistant, Mary Kubitschek, used the roller tube technique to place the cells into culture. It was observed that the cells grew robustly, doubling every 20 to 24 hours, unlike previous specimens that died out. The cells were propagated by Gay. This was the first human cell line to prove successful in vitro. HeLa cells proliferate abnormally rapidly, even compared to other cancer cells. Like many other cancer cells, HeLa cells have an active version of telomerase during cell division, which copies telomeres over and over again. This prevents the incremental shortening of telomeres that is implicated in aging and eventual cell death. The result is unlimited cell division and immortality. Contributions of HeLa cells to science. HeLa cells descended by cell division from this first cell sample are still being grown in laboratories around the world today because they are so much simpler to study than cells situated within the body. Cells grown in in vitro, that is, culture outside the body, have become an essential tool of cell and molecular biologists. In fact, much of the information that discussed today has been obtained using cells grown in laboratory cultures. Use in research. HeLa cells were the first human cells to be successfully cloned. HeLa cells have continually been used for research into cancer, AIDS, the effect of radiations and toxic substances, gene mapping, and countless other scientific pursuits. Polio eradication. Jonas Salk used the HeLa strain to develop the polio vaccine. Virology. HeLa cells have been used in testing how parvovirus infects cells of humans, dogs, etc. These cells have also been used to study viruses such as papillomavirus, HIV, herpes, mumps, etc. HeLa cells have also been used in the development of human papillomavirus vaccines. In the 1980s, Harold Sir Hussein found that Lacks cells from the original biopsy contained HPV-18, which was later found to be the cause of the aggressive cancer that killed Henrietta Lacks. His work in linking HPV with cervical cancer won him a Nobel Prize and led to the development of HPV vaccines that are predicted to reduce the number of deaths from cervical cancer by 70%. Cancer study. HeLa cells have been used in a number of cancer studies. HeLa cells were used to investigate the phytochemical compounds and the fundamental mechanisms of the anti-cancer activity of the ethanolic extract of mango peel, which was found to activate death of human cervical malignant HeLa cells through apoptosis. Genetics. Joe Intigio and Albert Levin, while working with HeLa cells, accidentally discovered and developed new and better technique for staining and counting chromosomes. They were the first to accurately describe that humans have 23 pairs of chromosomes rather than 24 as was previously believed. In 1965, Henry Harris and John Watkins created the first human-animal hybrid by fusing HeLa cells with mouse embryo cells, which would eventually lead to the Human Genome Project to study the impact of zero gravity on human cells. In the 1960, HeLa cells were sent to space to study the impact that a zero gravity would have on human cells, and it was discovered that the cell divided even faster in zero gravity. Laboratory weed. In 1967, works of Stanley Gartler found that HeLa cells had contaminated other human cell lines. The HeLa cell contamination had therefore brought to light an important culture contamination issue for the scientific community as a whole. This discovery was an important one, and it raised awareness and a need for better handling of biological samples to avoid contamination. Thus, HeLa cells have supported advances in most fields of medical research in the years since HeLa cells were isolated. Friends, I'm concluding this video here and see you soon in my next video. Thank you, friends.